Hereditary disorders. Almost every cell in our body contains a cell nucleus that holds 23 pairs of chromosomes that make up our genetic material. Each chromosome has within it the DNA, deoxyribonucleic acid, that makes up our hundreds of thousands of genes. Many of these genes make proteins that have a number of roles in the human body. Classification of hereditary diseases. They are classified to genetic diseases and chromosomal diseases. There are three types of genetic diseases. First, single-gene Mendelian disorders that are caused by mutation in one gene and are inherited by Mendelian laws. Second, multifactorial diseases that result from the combined effect of genetic composition and environmental influences. And the third is single-gene diseases with non-traditional, other than Mendelian inheritance. Chromosomal diseases result from structural or numeric disorders in chromosomes. Now, let us consider chromosomal mutations and chromosomal diseases. On the left, you see normal male karyotype, and on the right, normal female karyotype. Euploidy is the condition of having a normal number of structurally normal chromosomes. Euploid human females have 46 chromosomes, 44 autosomes and 2 X chromosomes. Euploid human males have 46 chromosomes, 44 autosomes and 1 X chromosome and 1 Y chromosome. Aneuploidy. Aneuploidy is abnormal number of chromosomes in the diploid set, non-multiple of haploid set. Xerosomy, absence of a pair of chromosomes, incompatible with life. Monosomy, absence of one chromosome in a pair. Polysomy are divided into trisomy, one extra chromosome in a pair, tetrasomy, 2 extra chromosomes in the diploid set, and pentasomy, 3 extra chromosomes in the diploid set. Mechanism of aneuploidy. Non-disjunction occurs when either homologs fail to separate during anaphase 1 of meiosis, or sister chromatids fail to separate during anaphase 2. The result is that one gamete has 2 copies of one chromosome, and the other has no copy of that chromosome. The other chromosomes are distributed normally. Monosomy. Monosomy is lack of one of a pair of chromosomes. Monosomy is most commonly lethal during prenatal development. A common monosomy seen in many species is X chromosome monosomy, also known as Turner's syndrome. X0 individuals are genetically female, however, they do not mature sexually during puberty and are sterile. Here you see manifestations of Turner's syndrome. Trisomy. Trisomy is having 3 chromosomes of a particular type. Trisomy is a specific instance of polysomy, a more general term that indicates having more than 2 of any given chromosome. A common autosomal trisomy in humans is Down syndrome, or trisomy 21, in which a person has 3 instead of the normal 2 chromosomes 21st. Down syndrome, or trisomy 21, has the following manifestations: characteristic facial features, short stature, heart defects. susceptibility to respiratory diseases, shorter lifespan, proneness to developing early Alzheimer and leukemia. Often sexually underdeveloped and sterile patients, usually some degree of mental retardation is present. Down syndrome is correlated with the age of mother, but can also be the result of non-disjunction of the father's chromosome 21. Here you see some manifestations or some characteristic features of Down syndrome. Now, Patau syndrome or trisomy 13. You see karyotype of a girl with Patau syndrome on the left and other manifestations of Patau syndrome. Edwards syndrome, trisomy 18. You see karyotype of a boy with Edwards syndrome. And the manifestations are almost every organ system is affected. It happens in 1 per 10,000 of live births. Children with full trisomy 18 generally do not live more than a few months. Non-disjunction of sex chromosomes, X or Y. There may be two variants of non-disjunction in males, which is in non-disjunction of sex chromosome in males is called Klinefelter syndrome. It may occur in two variants, additional X or additional Y chromosome. The gender is always male. Males with karyotype 47XXY have male sex organs, unusually small testes. They are sterile. They have breast enlargement and other feminine body characteristics. They have normal intelligence. Male with karyotype 47XYY are individuals that are somewhat taller than average and often have below normal intelligence. Klinefelter syndrome, please see the karyotype 47XXY. We see here tall height, feminine body build, gynecomastia, underdevelopment of testicles, and sterility. Trisomy X, karyotype 47XXX. These are females. It happens in 1 per 1,000 live births. Females are healthy and fertile and usually cannot be distinguished from normal females except by karyotype. Genomic mutations. Polyploidy is alteration in the number of chromosomes multiple of the haploid set. 3 haploid sets. Triploidy, 4 haploid sets. Tetraploidy, pentaploidy, etc. Polyploidy results from double fertilization and absence of the first mitotic division. Triploidy. A triploid individual has 3 of every chromosome, that is, 3 haploid sets of chromosomes. A triploid human would have 69 chromosomes, 3 haploid sets of 23. Production of triploids seems to be relatively common and can occur by, for example, fertilization by 2 sperm. However, birth of a live triploid is extraordinarily rare, and such individuals are quite abnormal. The rare triploid that survives for more than a few months after birth can also happen. Chromosomal aberrations, restructuring. Intrachromosomal aberrations include deletion, which means a portion of either the long or short arm of a chromosome may be lost, and this is termed a deletion. Then duplication, doubling of a fragment of a chromosome. And third kind is inversion, a rotation of a fragment of a chromosome by 180 degrees. Then interchromosomal aberrations are translocation, which is transfer of a fragment of a chromosome to a non-homologous chromosome, and reciprocal aberrations, which is exchange of fragments between two chromosomes. And you see the examples of interchromosomal aberrations to the right. Example of deletion is Cri-du-chat syndrome or cry of the cat, a specific deletion of a small portion of chromosome 5. These children have severe mental retardation, a small head with unusual facial features, and a cry that sounds like a distressed cat. Example of duplication, fragile X. The X chromosome of some people is unusually fragile at one tip, seen as hanging by a thread under a microscope. Most people have 29 repeats at this end of their X chromosome. Those with fragile X have over 700 repeats due to duplications. The syndrome affects 1 per 1,500 males and 1 per 2,500 females. Example of translocation. Here, a fragment of chromosome is moved or translocated from one chromosome to another, joins a non-homologous chromosome. The disease chronic myelogenic leukemia is caused by this translocation. The phenomenon is called Philadelphian chromosome. Single gene hereditary diseases. Single gene hereditary diseases are caused by mutation in one gene. Mutation in one gene, if it happens, it causes alteration in the sequence of nucleotides, which continues as derangement in the sequence of amino acids in a molecule of protein. In result, there occur qualitative or quantitative disorders in a synthesized protein, which finally result in different disorders in enzymes, structural proteins, transport proteins. Patterns of inheritance of single gene diseases. Autosomal dominant pattern. The diseases with this pattern of inheritance are called autosomal dominant disorders, and they manifested themselves irrespective of sex and type of carriage, homo and heterozygotes, and equally affect boys and girls. The extent of manifestations depends on penetrance of the gene. Probability for birth of an affected child is 50%. Characteristic is presence of the disease in each generation, which is called vertical transmission. The diseases may affect different organ systems. Please see the table which gives examples of autosomal dominant disorders. Huntington chorea. Huntington chorea is hereditary, slowly progressing disease of the nervous system, characterized by choreic hyperkinesias, mental disorders, and progressing dementia. Neurofibromatosis type 1 or von Recklinghausen disease. It is an autosomal dominant disease which happens at the same rate in males and in females. It is characterized by development of multiple tumors from nervous tissues. To the right, You see a picture with manifestations of neurofibromatosis type 2. Achondroplasia is a genetic disorder whose primary feature is dwarfism. In those with this condition, the arms and legs are short, while the body is typically of normal length. Other features include enlarged head and prominent forehead. The disorder does not genetically affect intelligence. Achondroplasia is caused by a mutation in the fibroblast growth factor receptor 3 gene. Autosomal recessive disorders manifest themselves only in homozygous condition. They do not depend on gender and equally affect boys and girls. They appear horizontally in pedigree, in siblings, brothers, sisters. Parents are usually clinically healthy, heterozygotes. The disease is provoked by marriages between close relatives. Examples of autosomal recessive disorders are given in the table below. An example of autosomal recessive disease is phenylketonuria. It is a disorder in metabolism of phenylalanine amino acid due to deficit of phenylalanine hydroxylase enzyme. The disease is characterized by accumulation of phenylketones in the body, which are toxic and induce disorders. Another example is galactosemia. It is a condition in which the body is unable to process galactose, the sugar present in milk. Accumulation of excessive galactose in the body can cause many problems, including liver damage, brain damage, and cataract. Alkaptonuria is a hereditary disease caused by loss of functions of homogentic acid oxidase and characterized by disorder in metabolism of tyrosine and excretion of high amount of homogentic acid with urine. The disease is characterized by disorders in musculoskeletal apparatus such as arthritis, osteoarthritis, of joint cartilages and soft tissues, and it is manifested by alterations in the density and color of soft tissues, for example, of ear auricles. Another disorder is damage to eyes, hyper or simply pigmentation of sclera, and damage to the heart, manifested by calcinosis of heart valves. X-linked recessive disorders, which usually affect boys with karyotype XY. Heterozygous females are carriers of a pathological gene and transfer the disease to 50% of sons, but themselves remain unaffected. All daughters of ill fathers are heterozygotes, and all sons are healthy. Father never transmits the disease to sons. The disease may occur in females, XX, provided they are homozygous for this pathological trait, or have karyotype 45X0. Please see examples of X-linked recessive disorders in the table below. It is an example of X-linked recessive disorders, Duchenne muscular dystrophy. You see the type of inheritance above and the clinical manifestations of this disease. The next example of X-linked recessive disorders is hemophilia. It is associated with deficit of natural clotting factors, which result in development of hemorrhagic syndrome. Now, X-linked dominant disorders. Since a gene is dominant, a disease affects both males and females. That is, it is not linked with sex. All sons of the unaffected father are healthy. All daughters are ill. An affected mother transfers a pathological gene to half of daughters and half of sons. The disease is milder in females. They are heterozygotes, than in males, who are hemizygotes. Examples include phosphate diabetes or vitamin D resistant rickets and type 1 orofaciodigital syndrome associated with multiple hyperplastic frenulums of the tongue, cleft lip, cleft palate, hypoplasia of wings of nose, asymmetric shortening of fingers. Now, single gene diseases with non-traditional type of inheritance. They include mitochondrial inheritance and Hollandric inheritance. Mitochondrial inheritance means transmission of a trait through genes of mitochondria. Cytoplasmogenesis. These diseases are equally transmitted from mother to both daughters and sons. Affected fathers do not transmit the disease to either daughters or sons. Examples include mitochondrial myopathy, cardiopathy, pigmented retinitis or Leber's neurophthalmopathy, mitochondrial encephalomyopathies, lethal infantile respiratory failure, syndrome of lactic acidosis. Hollandric inheritance is transmission of a trait from father to sons on Y chromosomes. Examples may be azoospermia, which means infertility, and growth of hair in ear auricles and on middle bones of fingers. Multifactorial diseases. These are diseases with hereditary predisposition. Hereditary predisposition may have different genetic bases. In some cases, hereditary predisposition is determined by one mutant gene, which is single gene predisposition. In other cases, hereditary predisposition is determined by a combined effect of several genes, polygenic predisposition. Criteria of multifactorial diseases. First, probability for expression of the disease in the relatives of the proband depends on the extent of their kinship with the proband. Second, risk of the proband's relatives depends on the number of ill members in the family. Third, genetic forecast depends on the extent of severity of the disease in the affected relative. Fourth, the extent of the hereditary load for the proband and its relatives increases if he or she belongs to a rarely affected gender. And finally, difference in the incidence in a family and a population, high in the family. Examples of diseases include essential hypertension, atherosclerosis, tumors, rheumatic fever, diabetes mellitus, psoriasis, schizophrenia. Disorders that show multifactorial inheritance. Please see them in the slide. Methods of examination of patients and their relatives. They include clinical genealogical examination, pedigree, family tree, twin method, that correlates the role of inheritance and the role of the environment in different groups of twins. Then population statistical method consists in examination of groups with different hereditary characteristics, nations or races. Cytogenetic analysis of karyotype and of sex chromatin. Biochemical methods, immunological methods of diagnostics, etc., and molecular genetic analysis, DNA analysis. Methods of prenatal diagnostics. They include non-invasive methods that are screening methods that can be used in examination of all pregnant females. They include ultrasonography in the first and second trimesters, maternal serum alpha-fetoprotein, chorionic gonadotropin, and of unconjugated estriol, which is done on the 16th to 20th week of pregnancy. Then, detection of intrauterine infection and isolation of fetal cells or DNA from maternal circulation. Invasive methods are associated with obtaining fetuses' cells. They include amniocentesis on the 7th to 20th week, chorionic villus sampling or of placenta on the 8th to 11th week, codosynthesis, which means taking fetus's blood from the umbilical cord, fetoscopy, and biopsy of fetus's skin. Thank you for your attention.